Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. I have an awesome guest for you today. Her name is Dr. Chiara Herzog. We talk a lot about her background and how she got started into this epigenetics world, as well as her being at the forefront of epigenetic research. We talk about recent breakthroughs that she's really excited about and why they hold so much promise for the future of healthcare. She historically has done work with epigenetics and the exposome and cancer risk and aging. So we break down all three of those categories and what her recent findings have shown. One of my favorite portions of this podcast today is reviewing her TirolGESUND study. The TirolGESUND study is actually looking at the longitudinal changes in the epigenome and other biomarkers of aging and disease in response to smoking and intermittent fasting. So this clinical trial has been running since about 2021, and they're retesting samples on these patients every 2 months. We also talk about her sitting on the executive committee of the Biomarkers of Aging Consortium and how we need more people to take steps to ensure we have a more comprehensive understanding of epigenetics and the role it plays in aging. We finally then end by talking about her current project she's working on and what she's super excited about in the realm of epigenetics. So just a little bit of an introduction to Chiara. She's a translational scientist, and she sits at the intersection of computational biology and clinical research at the European Translational Oncology Prevention and Screening Institute. She's also an honorary research fellow at the Institute for Women's Health at the University College London. Again, she's historically focused on epigenetics, other omics biomarkers as well, though, with the exposome, cancer risk and aging. And she's super passionate about leveraging novel technologies and biomarkers for risk prediction, early detection, and monitoring of personalized preventative strategies to extend health span. I mentioned she's co-leading one of the largest multi-omic human lifestyle interventional trials to date, that TirolGESUND study, and serves on the executive committee of the Biomarkers of Aging Consortium. And prior to her work, she obtained degrees in neuroscience and molecular medicine from the University of Edinburgh and Innsbruck Medical University. Now for my guest, Dr. Chiara Herzog. Welcome to the Everything Epigenetics podcast, Chiara. I'm excited to have you here today.
Thank you so much for the invitation. I'm glad to be on.
All right, so I just gave everyone the most amazing introduction I think I've ever recorded. So let's jump in and get this conversation started. This is the first time you and I have actually met and are chatting. As I mentioned, you're a translational scientist, an honorary research fellow. Your work focuses on epigenetics, a lot of other omic biomarkers. And what I think I'm most excited about to talk with you today is that you're one of the co-leading authors of the largest multi-omic human lifestyle intervention trials to date. You're even on the executive committee of the Biomarkers of Aging Consortium. So just, whoa, I want to talk about all of these things. I'm super impressed with everything that you're doing. And I know I even mentioned during that introduction, before all of this work, you actually got your degree in neuroscience, but I know that just can't be the whole story. So how did you even get into all of this? Where does it start? Give us all of the information.
Yeah, so that's a good question. I think it starts and ends with curiosity, first and foremost. So I think that has led me through my path so far. Both as a scientist and a person, I just love, learning things and figuring things out. And I've always been really interested in finding out how diseases happen on a cellular level. And I actually got my undergrad in molecular medicine, so that's kind of 2/3 normal medicine, and then the other third was more research focused, so genomics, bioinformatics, all of that. That program was kind of designed for people like me who didn't necessarily want to become medical doctors, but wanted to work with kind of medical research and patients and all of that. So at the time, that obviously was a really great foundation. And then I became super interested in finding out how we regenerate, because humans actually aren't very great at that. But other model organisms are, like zebrafish and axolotls can regenerate their limbs and brains and spinal cords after an injury. And I was like, okay, I need to figure out how this works. So I did pursue a master's in neuroscience and a PhD in neuroscience from the University of Edinburgh, who are really focused on regeneration and neuroscience. And during my PhD, I did very different things than what I do now. I worked on zebrafish, brain injury repair, and kind of factors that make the brain permissive to repair, which is really exciting. So they can really functionally repair their brains using microscopy and RNA-seq and CRISPR and all of these things. And yeah, while I did really enjoy the work and found some interesting stuff, I did want to take a step closer to kind of my basics of like, I want to work with like really close to patient research, which then led me to an omics precision diagnostics company. And during that time, I coincidentally met my current mentor, Professor Martin Lichwenter, and he had been working on epigenetics for 25 years. When I spoke to him and he explained what he does for his work, I was like, wow, okay, I need to do that. I'm convinced, like, how can I get involved? And I think, yeah, this is kind of when my whole like research into epigenetics started. And I'm very glad that I ended up here.
Yeah, no, that's a great story. It's interesting too, because, well, I guess it's a theme and a lot of the hosts I have on the show, it's like they just kind of ran into epigenetics at some point in their career. It's not like they started off studying that. And I'm the same exact way. I had no idea really that field even existed. Like 5 years ago, I think I was taught maybe one day in traditional like university school about epigenetics and maybe the difference and how it compares to genetics, but that's about it. So I appreciate you sharing the story and the curiosity section. I actually have a sticky note on my laptop that says, you don't have to be right. You just have to be curious. So I love that you said that, our ability and willing to learn
Yeah.
and open up to this as well.
Yeah. And
so
the other thing, oh, sorry.
Yeah, go ahead.
The other
go
thing
ahead.
I wanted to say, like, I'm actually really glad that you're doing Everything Epigenetics, because almost everyone to this point has heard about genetics and like, you know, we all learn it in school. And then once you start talking about epigenetics, people are just like so confused and they're like, wow, this sounds really scary and I don't know what it is. So I'm like, you know, we need better education, even within like biology and medical research. We need to spread the word.
Yeah, definitely. No, I'm 100% with you there. So thank you. I appreciate it. And again, you do a lot of interesting work. When I was creating this kind of agenda and what I wanted to ask you about, I was thinking, okay, well, I want to start broad. I know you've been at the forefront of a lot of different areas of epigenetic research, but I want to know and hear from you directly just what recent breakthroughs are you most excited about? And then really secondarily to that question, why do you think they hold such a promise for the future of healthcare?
Absolutely. So I'm sure you and your community will know that there's lots of things to be excited about epigenetics. One of the things why it's so powerful is that it can integrate genetic and non-genetic factors. So it gives us more information than if we only look at genetics or epidemiology. And the other exciting thing is obviously that it's dynamic, so we can potentially mitigate or reverse some of harmful effects. And this is, I think, quite a hopeful message for taking charge of our own health. Every day nowadays, we're really learning a lot more about how exposures and maybe the aging process modulate our epigenome, thanks to very vibrant and active research communities. So I think that's pretty exciting. We're also able now to leverage knowledge that we already have about epigenetics to study the impact of new exposures that maybe haven't been around for so long. So for instance, I've recently looked at the effects of e-cigarettes they've only been around for maybe like 10, 15 years. And the long-term studies are kind of lacking. So we can at least use epigenetics to estimate those risks. More broadly speaking, I think we can really estimate these biomarkers with epigenetics, maybe also other biomarkers, but obviously epigenetics is a great candidate for that to estimate the impact of health span extending interventions. So rather than having to wait decades for a clinical outcome, we could maybe use an epigenetic biomarker to much quicker assess the impact on biological age and possibly down the line, the kind of health span risk. So yeah, I think in the future, I would almost imagine that routine epigenetic testing could be used to personalize preventive strategies, but also guide more targeted intervention before a disease develops, and rather shift the healthcare system from being reactive as it is now to a more proactive system to give individuals control over their own health.
Yeah, absolutely. And if you had to, let's just say, guesstimate as well, how far do you think we're away from that? I mean, that's what I spend most of my time doing at True Diagnostic, right? Is working with healthcare providers, but definitely the healthcare providers that aren't seeing the everyday patient. They're seeing people who are really optimized and healthy. And I think one of True Diagnostics' goals and my personal goals is bringing this testing out to the masses, just because we know how important it is. And it's the people who, probably have those more poor socioeconomic statuses or associated with, worse epidemiological factors. They probably need this testing the most, right? We can really affect outcomes of their health. So how far away do you think we are in maybe getting some of this into more of the clinical picture, coming from like a research standpoint?
Yeah, absolutely. Yeah, this is a really great or important point to talk about. Unfortunately, I think we're not quite there yet, although I do agree there's promising progress being made. One of the challenges in today's health systems, and as I'm sure you know as well, is that we're still very much a sick care system rather than a healthcare system. So I think we really need to move away from that only looking at disease, but rather towards maintaining health and kind of extending health even, which then can allow people to kind of stay more healthy for longer. Just to put that into context, in terms of like prevention and spending on treatment of disease, both in the US and Austria, which is where I'm from, the total or the proportion of healthcare costs that's spent on prevention is about 3%. So it's really not a large amount. So we need to change that. And I do think that kind of these tests that we develop, either epigenetic or other tests, could kind of serve a key purpose in towards maintaining health. The issue is there at the moment that a lot of them aren't as affordable, and as you say, like there's kind of a bias in the sense that people that need them most maybe aren't reached. So I think what we really need to do is kind of generate some regulatory evidence and also enable reimbursement, so these tests become free or very affordable for people. What we've also seen is that at-home testing and kind of self-sampling can help to increase participation in populations that maybe are typically underserved because they maybe don't regularly engage with a healthcare provider. So for instance, I think in cervical cancer screening and also lung cancer screening, people have piloted just sending sampling kits to people at home, and they found that actually increased participation. So I think we really need to make these tests more widely available. The one thing that I do agree with you is that you mentioned people, that are typically from lower socioeconomic background are maybe less likely to engage. And there's like an additional thing to be said that I'm really excited about the prospect of being able to give more or let people take more charge of their health, but also we shouldn't place all of the burden on the individual. So I think the healthcare systems really still need to create this permissive environment where people are supported to their needs as well.
Yeah, there has to be some type of plan in place to distribute the testing, then also have the follow-up care associated with it. I think that's super important. So with all of your work and studying epigenetics and the exposures, I have to ask a personal question. What have, you mentioned the e-cigarette example, like with all of your studies, have you done anything or changed anything personally from learning how different exposures affect the epigenetics? I know I've spoken with some researchers from this field, and they talk a lot about like air quality and water pollution, and they've actually changed things like in their own house to help their epigenetic expression levels. But what about yourself?
Yeah, absolutely. So I mean, I have to admit I've always been kind of healthy in a sense. So I've always been very focused around exercising and diet and all these things. So I think maybe that's also why I ended up in this research to kind of show, we have the data, how this helps. But definitely there's a lot of really interesting stuff on socioeconomic status and where you live, if you live near a green space or not near a green space, this influences your health. So I haven't actively done anything yet, but I'm obviously very health conscious and always keeping it in the back of my mind.
Yeah. That's I know I love that paper with the more green space, having a positive effect on your epigenetics. So it's always my excuse to go and buy more plants. And to fill my house with plants. Awesome. So the next topic, Chiara, that I really want to talk about is the study that you're co-leading. So this is what I'm super excited to learn about. I wasn't super familiar with this actually until diving more into your individual work. So what is the study called? How do you pronounce it?
It's called TirolGESUND. Yeah, so Tirol is the region where we're based, and Gesund it actually means healthy, but it's like an acronym for what the study is now.
Awesome. And I know with this study, you're looking to really assess longitudinal changes within the epigenome, which is extremely important, along with a lot of other biomarkers of aging and disease in response to, I think, smoking cessation and intermittent fasting. So I'd love for you to just maybe set the stage for this trial, and I'm sure I'll have some more follow-up questions about it too.
Sure. So as you said, this is kind of one of the first studies to also really focus on epigenetics as kind of a primary outcome. Because there have been studies where they did look at epigenetics as well, but we really set this study up as like, okay, we want to see what happens to epigenetic biomarkers over time. So we recruited over 150 women and asked them to either do intermittent fasting or smoking cessation. Both of these interventions we do think are at least health promoting, and especially intermittent fasting possibly health span extending, although it's not been confirmed in humans yet. And at the center of this study is really we take samples every 2 months from different tissues, so blood, cheek swabs, and cervical samples, and are following biomarkers over time. So obviously there's already really exciting different epigenetic biomarkers in each of these tissues actually, but we don't really know how dynamic they are over time, or at least it's not been super well characterized, and also how they agree with each other or maybe disagree. So there's like hundreds of epigenetic clocks, I would say, at this point. And we don't know, like, are they going in the same direction? What's happening? So I think this is kind of a study that we want to start to really benchmark these biomarkers across different tissues. The other thing that sorry, you go ahead.
No, go ahead.
The other thing that I'm quite excited about is that we're not just looking at epigenetics. So one of the things that's also often a little bit criticized when talking about epigenetic clocks is that we're like, we don't know what it means, and like, what does it relate to even in like terms of a healthcare association or outcome. So we won't have outcomes because the study has only been started in, I think, 2021, and we've only followed the participants for a maximum of like 3 years. But what we're trying, so we really deeply phenotyped these participants. So we did a bunch of like clinical routine tests. We also did functional exercise capacity, immunome, microbiome, metabolome, all of these things to really gain a better understanding, like what do epigenetic biomarkers actually associate with, and can we leverage this in the future to refine biomarkers or kind of at least catalyze future research with this kind of data collection.
Yeah, it seems like a really well put together and thought through study too, grabbing other additional biomarkers to see how they're correlating with the epigenetic age clocks. So yeah, a couple follow-up questions there. I'm all for this study. I would love to take part in it as well. You mentioned that there are only women, correct?
Yeah, that's right.
Yeah, absolutely love it. I think there's a lot of data. I think there's a lot of or underrepresentation within women in these type of clinical trials. But why just women? I'm just curious on that.
Yeah, so our institute's original focus was very much on women's cancer. So we've been doing a lot of cancer biomarkers as well. And obviously, the study originally, when we started thinking about it, was more about cancer prevention. And then I thought, well, actually, a lot of these steps, especially intermittent fasting, are cancer preventing, but also kind of possibly gerotherapeutic. So why not look at everything? So the original focus was because of that focus on women's cancers. But we have actually now we have follow-up studies from the TirolGESUND study where we've recruited 600 people, including men and women. So hopefully we'll get data on both genders in the future.
Good. Yeah, I saw on the website as well, you had a vision by 2040 to make women's cancer a disease of the past by hopefully. Then too. So yes. that definitely makes more sense. And then, Yeah, so I think a lot of listeners, are probably familiar with like the calorie randomized control trial that's out there. Really amazing study. It's that 10% overall caloric restriction in healthy non-obese adults over a 2-year period. So that's kind of like the one recommendation we can give to people. That's really peer-reviewed, published. There's a lot of scientific evidence behind it. But you and your team, you're looking at intermittent fasting. What does that intermittent fasting schedule actually look like?
For our study, we actually left it pretty open to participants, which has pros and cons. So obviously you want to have like very tight control over what people do, but they're still people after all. So you can't really tell them too much what to do. But we did a 16 to 8 fasting. So that's basically 16-hour fasting a day and then consuming food during 8 hours. And that's usually pretty achievable for most people. And actually a lot of people already do that subconsciously if they don't have breakfast. So indeed, in that study, we had a lot of people who just skipped breakfast. Actually, during our study, we also read another paper that was super interesting. It was in mice, but they showed that if you align your circadian rhythms with the time that you fast, then actually have greater benefits. So in translating that to humans, it's actually better to skip the dinner than the breakfast, which is very annoying because it's much harder to skip dinner. But that is what we actually looked at in our next study. So in the bigger study, we actually specified that people should have their last meal around 4 p.m. In that first study, we didn't specify it, but we did see quite good effects. So the studies are still undergoing peer review at the moment. But a lot of the participants did lose quite a lot of weight, even though that wasn't really the goal of it. Obviously, that's still a benefit. So we did see quite a lot of sort of beneficial changes as well, at least from a clinical perspective as well.
Sure. Yeah, that makes sense. And yeah, I actually like the skipping dinner part. I love breakfast, but if like a lot of people still work right at 4 o'clock, so that's where it becomes really hard. Is like I'd have to bring food and like cook it at work or do something of that sense. So I think I heard that you maybe presented some of these results at the Gordon Research Conference as well.
Yes.
Are you able to give any insights into what you saw from like an epigenetic standpoint? And if not, that's also totally okay.
Yes. No, it's fine. I mean, we're still, as I said, undergoing peer review. So hopefully these findings will be published soon. But we did see some changes, but they're not all easy to interpret. So especially looking at a couple of different epigenetic clocks across the different tissues, it wasn't, as straightforward as one might expect. And actually, I wasn't expecting that because all of these clocks are trained differently. My hope is that this is just like presenting what we found, but the idea would actually be that we can leverage this data set to improve our understanding. So that's kind of my main focus now, to see what do these epigenetic clocks actually correlate with in terms of like more interpretable features like clinical changes or maybe immune cell changes. And then we could actually learn more about them, which I think is quite exciting.
Yeah, absolutely. Well, hopefully by the time this episode is published, when the episode is published, the paper will be published and we can link it and can give listeners a little bit more information because it's definitely a hot topic in the space right now. I think I see all of these researchers coming out and saying, intermittent fasting doesn't work, or it does according to which biomarkers you're looking at. So I'd love to learn a little bit more about how they're working from an epigenetic standpoint. So
absolutely. Yeah, there's a lot of interesting evidence actually on like a more cellular level of like intermittent fasting basically activates autophagy and all of these things. So that's not something that we looked at in our study, but hopefully one day there will be biomarkers of autophagy in some way that we can look at.
Yeah, I've never even like that hasn't crossed my mind as biomarkers of autophagy. But if you could even measure that through DNA methylation or epigenetics, that would be super interesting as well, because I know we're trying to capture some like even senescent based markers through epigenetics. So that's an interesting thought process. All right, awesome. So thanks for that amazing introduction to the TirolGESUND study. I kind of want to skip around and maybe talk about your work focusing on the association of epigenetics and cancer. So I think connecting maybe what you said with your goal of the research institute and focusing on, making women's cancer a disease of the past by 2040, you have some work with cancer risk and aging. So given your expertise there, how do you see epigenetics being used effectively to diagnose cancer or maybe even predict cancer risk? I know there's some of that stage 1, stage 2 cell-free DNA methylation testing within cancer. Can you talk a little bit about that?
Yeah, absolutely. So, as you've mentioned, there has already been quite a lot of work in that space, especially for detecting cancer. And this is mainly because the epigenetic profile of a cancer cell is so disrupted that it's actually quite easy to tell apart from a healthy cell. And this is what a lot of companies like Grail are using to detect DNA methylation of cell-free DNA, which the tumors shed into the bloodstream. And then they can basically tell if a cancer is there, and if so, where it is. One of the challenges with that approach is that although it can detect some stage 1 cancers, it actually works much better if the cancer is bigger and sheds more tumor DNA. So this is a little bit problematic also because there's different tumor types that shed different amounts of DNA. So we actually found that for some cancers, including endometrial or womb cancer, other sample types might be better. So we recently developed a test that's leveraging DNA methylation in cervical samples that are already used for cervical cancer screening to detect endometrial cancer tumor DNA. And this is now an epigenetic test that has been clinically implemented in multiple European countries. So that was quite nice to see that we can actually apply this. However, the one challenge, if we look at tumor DNA, regardless of whether it's with liquid biopsy of blood or other sample types, is that per definition, we already need to have a cancer present. So coming back to your original question, after a long detour, one of the things that I'm super interested in is how can we use epigenetics to actually predict disease before it develops? And one of the things that we've been working on in that space is to leverage what we call so-called surrogate samples. So they're epigenetically similar to the tissue at risk, which would be a tissue that might develop tumor inside the body, but the surrogate tissue is much easier to access. So we've been using cheek cells or cervical samples, which contain sources of epithelial cells, which are similar to what cancer cells or some cancers might develop from, to read out kind of what's the epigenetic state. So we don't have tumor DNA in those samples, but the kind of epigenetic footprints of exposures and aging and other things might still be similar, even though those surrogate samples don't develop cancer themselves. And with that approach, we actually found that we can detect the risk for multiple women's cancers and also lung cancer, which isn't a women's cancer, but somehow ended up in our portfolio years in advance of a diagnosis, possibly early enough to intervene. So I'm really excited about this, and I do think this could change the way of how we think about cancer prevention, but obviously there's more research and validation needed for that.
Yeah, of course. No, that's super interesting. You mentioned the endometrial DNA methylation testing that's already like used in clinics and whatnot. Do you mind mentioning the name of the company? I don't think I'm familiar.
Yeah, so it's called Sola Diagnostics, and so this is something that we kind of spun out as part of our research because obviously there's a lot of research that is done in academia but doesn't actually make it to a patient. So that was really important for us to enable translation of that, and yeah.
Awesome. Thank you. No, I'll definitely look into that a little bit more. I think it goes to our kind of conversation from the beginning, which is, how quickly can we get this into the healthcare system? And I think you all, that's a great example of how you took your research and you branched off and you created that into something that providers can use in clinic. But if you never have that step, then you're going to miss that translation and it's never going to get to the end patient. Yeah. Awesome. And you said, I think you mentioned one more, you said endometrial cancer. And then what was the other type of cancer where epigenetic markers were proving to be really promising?
Yeah, so we actually had we looked at breast cancer. Okay. So we already have a publication for cervical samples, but there's another publication coming out soon where we show that we can actually detect breast cancer with a cheek swab, which is still mind-blowing to me, but very cool. And also lung cancer. We've also worked on other women's cancers, including ovarian cancer and cervical cancer.
Got it. Thank you for the clarification there. All right. I want to move on to another topic here as well. So you're on the executive committee of the Biomarkers of Aging Consortium. So I know you're actively promoting a lot of standardization for research into biomarkers of aging. I think right now epigenetics is really heavily used in that, but we want to see what other kind of omic markers can be useful to study aging. So with that being said, I was actually at the first Biomarkers of Aging consortium at the Buck Institute last year, which was awesome. I know you all are having the second annual meeting at Harvard this year, which I'm so sad I'm not able to make it. I'll be traveling for another conference. But all that to be said, I know you also have a special interest in like employing more robust methods to advance molecular biomarkers into those promising clinical tools for personalized disease prevention we keep coming back to. So tell me a little bit more about your role there and what the Aging Consortium is, what your goal is. I'd love to hear a little bit more in detail from you.
Yeah, sure. So we were actually founded in 2022 at another scientific meeting. I think it was the Cold Spring Harbor Mechanisms of Aging. And at the time, there was kind of a push in the community to really come together because everyone was developing biomarkers, but everyone was comparing them separately. And we had no idea, which ones actually work. We're not speaking the same language. Can we bring some sense into that? So since then, I've been working very closely with other researchers, including many at Harvard and Stanford, to drive consensus. Our kind of, I think, success formula was that we tried to bring together lots of experts rather than trying to go at it on our own. And we've been working on a couple of different things. So on one hand, we want to build roadmaps for the community to either, for instance, speak the same terminology. So that was one of our first manuscripts to kind of define what is a biomarker of aging, which is a surprisingly challenging concept to agree on. And other things like how can we kind of bring them in line with what FDA classifications of biomarkers are. We've also worked on kind of a review of current validation efforts and what's needed to be next. And our most recent work, which I've just found out will be published on Monday next week, so the 16th of September, will tackle a lot of the challenges that hinder the clinical translation of biomarkers and recommendations on maybe how to overcome them. We're also, as you mentioned already, hosting scientific meetings to bring together the community, and it's really sad that you can't make it, but hopefully you'll be able to come next year. So yeah, our meeting will be November 1st and 2nd at Harvard Medical School this year. There's actually something else that's exciting that's starting at that time. So we're initiating our own biobank and kind of longitudinal sample collection. And the sample collection will actually start on site at the meeting in Harvard. So yeah, that should be quite exciting. So it seems like we weren't stressed enough by the conference alone. We added something on top of that. So yeah, I think that'll be quite fun. And we're really excited to build open source tools for the community. So adding to kind of standardization efforts, we built this open source library called BioLearn. So it's really easy to compute biomarkers. I think most of them are epigenetic actually. And we have 30 that are in there at the moment, and it's still growing. And we've also just generated kind of the gold standard methylation data set that people can leverage for benchmarking of aging biomarkers. So yeah, I think we're trying to kind of organize the community and support the community in a way that we can catalyze research.
Yeah, that all sounds amazing. It sounds like there are multiple different initiatives. I'm interested in the sample collection on site. I think that will be a really good like cohort or biobank, you can call it, because it's going to encourage the same people to come back, right, and start gathering longitudinal data, which is the big need here, especially saliva-based longitudinal samples. I know that really aren't available in any biobanks that are created today. So super exciting. So I'm also very excited about that paper that you said will be published on Monday after this. So congratulations there as well. I think anything to help me and support my cause on proving to healthcare providers how clinically important this testing is, even though it may be a little bit hard to interpret at the moment. I think it's more of a larger, data play for future analyses even, but still being able to collect a sample and create almost like their clinical biobanker cohort now, so to speak. So is there a consensus? Like you mentioned, most of the aging biomarkers and kind of what's available is like epigenetic based. Again, I obviously like that's my passion and the topic of the podcast, but I would be super naive to say that that's the end all being. We also need to grab proteomic markers, and there's some pretty great proteomic based clocks out there, even transcriptomic based clocks and some others as well. So where do you think that consensus really is? Or is it just, and do you ever think we'll really decide, or do you think it's just kind of getting closer and closer to the truth, which essentially that is science? I think there'll always be conversation around this, but is there any way we can start to clear the air here?
I think we can, and I think the answer lies in what you've mentioned before with collecting samples and data. So one of the challenges that we face, and this is also something that will be discussed in our paper, is that we need to really benchmark under the same conditions, maybe even the same population, which biomarkers actually work the best. And I think only the data can really give us the answer because, as you say, obviously I'm super passionate about epigenetics, but I'm not, I'm not denying that other biomarkers might be might have great potential. One of the challenges also here is that there is data, but it's often very hard to access. So, it can be very time consuming and challenging to even get access. And then once you have the data, you still need to clean it and pre-process it in a different way. And it might be kind of, chaotic. So we need to have kind of standardized data formats, standardized data collections that can really facilitate this benchmarking. And I think this will be quite important. The other thing is that I think we're still not really talking enough to clinicians as a community. And this is something that we actually want to tackle in one of our next projects, because we need to understand what do clinicians think about this and how can we tailor biomarkers to really support clinicians. So yeah, if anyone's listening or knows clinicians, please do get in touch, because we want to hear from you regardless of whether you're an expert in epigenetics or not.
Yeah, definitely. And we'll talk about that after the off air too, since we have,
For sure.
all of our healthcare providers and kind of their cohorts as well. So, no, that's super interesting. Now, the paper you all are publishing on Monday, I know I'll have to wait to read it, but you're suggesting that idea of like doing the benchmarking at the same time, like gathering the longitudinal samples. Is that correct? Or do you all actually have that data already?
No, so that manuscript was mainly to identify what are the kind of key roadblocks that exist today and how can we overcome them. So we hope that this will be, this will kind of highlight some focal points that we need to tackle as a community, and not just the community. I mean, it's also basically not just up to us, but also collaborations and funding systems to really allow to have that happen. So of course, people want to measure biomarkers or maybe want to collaborate with clinicians, but if there's no funding allocated for that, then it will be quite challenging.
Of course. Yeah, absolutely. And we talked about this a little bit. You just mentioned you're gathering and grabbing samples at the next conference, like actually in person, which, as logistically crazy as that might be, I think it's the best way to go, because, you ship people a kit or you get them excited about it, and then their kit just sits on, their kitchen table for days, and it's harder to get that returned. But are there any like other steps or things that you think need to happen to ensure that we have a more comprehensive understanding of epigenetics across diverse sample types? I mean, this is kind of what you did with the like cancer prediction tools and whatnot. Is there anything else we can do, even just besides like education or sample collection regarding that?
Yeah, I mean, there's a lot that still needs to be done in that space, because we know that obviously most people focus on blood because it is the most easily available. Other sample types can be more, if not, or equal, if not more informative. So we do need to learn more about this. I think currently there's not a lot being done, and it's just the challenges of sample collection and getting the population. So obviously, if you had an ideal cohort, you would try and collect everything, but then that's not going to work. So you need to be kind of make it feasible enough for participants to still come back to you and give their samples.
Yeah. Yeah, absolutely. All right. So a couple more questions. We're kind of getting to the end here as well, Chiara. We hinted on this, I think, a little bit and continues to be the theme of the podcast, but really looking ahead, let's imagine maybe way farther ahead, maybe even 5 to 10 years or so. Um, what do you think epigenetics will look like, and what can we do now? Like, what are the next critical steps for really advancing the use of epigenetic biomarkers and clinical research? This kind of goes back to the gaps in the current research that you would
Mm,
like to be addressed. But is
yeah.
yeah,
Mm. It's challenging. So I think, again, it does all start with data. So one thing that I am quite passionate about is trying to have a sort of data set that are enabling data access for people while maintaining participant security. That's obviously very important, but this is really a major roadblock to a lot of research at the moment because we need to share data to understand more. And if it's all siloed away on one person's hard drive, it's not going to benefit the community. So that's something I think that will help us understand more. And then obviously the other thing is really this relationship with clinicians to understand under what circumstances can these biomarkers be used, maybe initially to select participants for studies, maybe to make healthcare decisions in the future once they've been validated to do so. So I think this is more like a gradual progression, but we need to lay the foundation really solidly now. And yeah, have the right evidence as well.
yeah. I think on a personal experience with me, like spending all this time speaking with healthcare providers, it's a little nerve-wracking, right? To be able to say, hey, let's share, their patient data, their cohort that they've worked for their entire lifetime, right? So I wonder if there's, the ability to obviously share that data in a way that's obviously super secure, which happens with any type of data transfer and research anyways, but also a way to highlight the healthcare provider, right? And the individual and show the work that they've done, whether it's in terms of like them being able to say they changed those biological age clocks, hopefully in a positive way, and even publish or do some white papers for those particular clinics. I think any type of publication is really interesting to the healthcare provider for them to be able to use to market their work and show that they're a leader in the space when really being able to take patients' health in their own hands. And again, these aren't your typical clinics, right? These are your clinics who are doing more like healthy, longevity, optimized based medicine. But that's usually the feedback I hear from them. So I'm wondering, and I usually have a lot of healthcare providers who listen to this podcast as well, if there's a way to help bridge that gap and again, get them more involved. So will be
Yeah,
a topic that we'll
sounds
have to
great.
continue
Yeah,
for sure.
no, that sounds great. And I absolutely agree. You need to have some incentive and also recognition for the people that are, sharing their work with you. So it's not like they need to go uncredited. I think it's quite important. Also, the individual participants, because, they're actively contributing in research. So this is extremely valuable. And we always say it couldn't happen without the participants as well.
Right. Yeah. That's something to think about is, hey, we can go to the provider level, we can get them involved, right? How do we even go down to the patient level to hopefully continue to get them to test longitudinally too? All right. Another question here, Chiara. Can you share a bit about what is like your main focus today? Like, what are you spending your day to day working on? That's the first part of the question. Second part being, are there any other upcoming projects that particularly excite you in the realm of epigenetics?
Yeah, so my day to day is pretty chaotic because there's many different projects. I'm trying to keep track of them. But there is some interesting stuff coming up. So one study that I'm kind of tying up at the moment is looking at tissue aging across different tissues with epigenetics. And not unexpectedly, we see that, for instance, cancer tissue has a higher biological age than normal tissue. So specifically looking at the breast. But one interesting finding that we had is that some tissues from cancer patients actually show a lower biological age than controls. So if we look at a cervical sample from a breast cancer patient, they show a lower age. And this really surprised us. So we actually went ahead and repeated this in a mouse model, and we saw exactly the same thing. So to us, this indicates that we do need to dive deeper in understanding, okay, what do we actually measure here? And this is through a range of different clocks. And how can we interpret these findings, because typically you would say younger biological age, great, I've rejuvenated, or I kind of extended my health span. So I think this is quite exciting. So we don't quite know what it means yet, but hopefully this, I think most discoveries in science, you stumble upon something unexpected, and then that's actually where the real discovery starts.
So the other, I want to hop in there too, Chiara. Any hypotheses as to why? Like that's, I hope everyone who's listened to this so far has like stuck around, because now I'm like, oh, this is super interesting.
No, I think we don't really know yet. I think there's probably, there's one hypothesis that we have, is that there is some systemic effect that obviously is affecting different tissues across the body differently and predisposing to maybe cancer formation. But we don't have any way of proving that at this point. So this is future work. But I think this should be super exciting. And also digging a bit deeper, because obviously most of the biomarkers today are not, we don't really know what exactly they're measuring in terms of epigenetics. So I think that would be quite interesting to follow up as well. And maybe it's like only certain things that are being rejuvenated and not others.
Right. And is that being done in like first generation clocks, second generation clocks, kind of like a full platform?
So, we've done a bunch of different things just because we wanted to have like a broad comparison. We've also just kind of developed some functionally enriched clocks for certain hallmarks of cancer and aging, including kind of senescence and proliferation. We do see this kind of across the board, which is was very surprising. But hopefully that one should be out soon as well. I'm also, as ever, expanding some epigenetic signatures of exposures. So we've previously looked at smoking and viral infections and all of these things. So we're complementing that to really get a better grip at what do different epigenetic markers actually mean. And lastly, I think I've already touched upon this, where we have these like large follow-on studies in humans, where we have, I think in total, over 700 or 800 people that we followed over 8 months and ongoing. And we have longitudinal samples from them. So that will be really interesting once the data come in.
Yeah. Well, awesome. No, that all sounds great, Chiara. Thanks so much for giving us a little window into like your chaotic day to day, but then also long term as well. All right. One last question. This one's a curveball. Chiara, I ask all my guests this. If you had to pick any animal to be, what would you choose and why?
I would choose raccoon, because they're chaotic and clever and have great hands for fast typing on
and you're cute too. I don't know if you watch any of the like Guardian of the Galaxy movies with the little raccoon in it. What's his name?
Rocket.
Yeah, Rocket. He's super cute. I haven't had anyone say that so far, but it sounds like you thought about it because you knew.
I did not know that, but they are my favorite animals. So
very cool. Well, awesome. Chiara, we've come to the end of this really awesome podcast interview. I had a lot of fun talking with you. If anyone wants to get connected with you, where can they find you? I think you're on Twitter and some other places as well.
Yeah, absolutely. So all of my information is also on my website, which is chiaraherzog.github.io, or just connect to me on LinkedIn or Twitter, or you'll find me.
Yeah. Awesome. Well, thanks so much again. I really appreciate your time. Everyone who's listening, thanks for joining the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more about how. Thanks, Chiara.
Thank you so much. It was great fun.